Good morning, everybody, to this first set of presentations of the 2022 Cornell Nutrition Conference. My name is José Santos, and I will be presenting here in the program of BioChem, New Revelations on Transition Cow Nutrition. We just heard from Dr. Mike Van Amber what makes nutrients require in the diet of dairy cattle. And I hope that I will make the case today in my presentation that choline fits those criteria of a required nutrient, which we know it is required for every mammal that's being studied on, but not really clearly dissected in the case of lactating dairy cows. So during my presentation for the next 38 to 40 minutes, I want to tell you a short history on choline, and then I will show you some of the roles of choline on intermediary metabolism. I want to present some data on the role of choline in reducing hepatic lipidosis in dairy cows, and some new data showing that, as in many other species, choline also has this role in dairy cow. I'll spend a little bit of time talking about the impacts of supplementing choline during the transition period and the effects on postpartum productive performance and a little bit on health. And then I want to make the case that every cow during the transition period benefits from supplemental choline in the form of rumen-protected choline. And then lastly, I just want to close with some potential mechanisms. Some are already well established, but others will be hypothetical based on data from other species. And when we pull it all together, I hope to make the case that choline is a required nutrient for ruminants, and it should be supplemented in the diet of transition dairy cows the same way it is supplemented in the diet of pre-ruminant calves. So choline was discovered in the 1700s by French and German chemists and pharmacists. But it wasn't until the early 1900s, during the discovery of insulin by Frederick Banting in Canada, that one of his students, Charles Best, after he completed his studies in Canada, he came back to the US and then also worked in Toronto, that during his studies on depancreatized dogs, that he showed that those animals would develop hepatic lipidosis. So when dogs are removed the pancreas to study the role of insulin on glucose metabolism, this induces diabetes in dogs, obviously. Yeah? And during diabetic states, animals have hypertriglyceridemia, and also insulin, which would be devoid in those cases, is important to stimulate the secretion of very low density lipoprotein. So one of the consequences of removing the pancreas that was observed by Charles Best was that dogs would develop hepatic lipidosis for those reasons, an excess of synthesis of hepatic triacylglycerol concurrent with the inability to export them from the liver to the systemic circulation. But what's important here was that he eventually showed that supplementing phosphatidylcholine or choline in the diet of those dogs, he would be able to partially reverse hepatic steatosis. And this is what set the stage for the subsequent discoveries on choline in the monogastric literature and eventually in the human literature to demonstrate that it was a required nutrient in the diets of every mammalian species that's being studied. But the interesting thing that it took almost 70 years for that to be established in the human literature for the recommended dietary intake of choline to be established for humans. So, and it was because of the work of several scientists, including Steven Zeisel from the University of North Carolina, who showed that choline played an important role in brain development and cognitive function, particularly because choline is an essential component of phospholipids and is an essential component of sphingolipids. So obviously that plays an important role in every cell, but including neurons. But then when we move back to the ruminant literature, it was in the early 80s that it was shown with sheep models using radiolabel tracers that most choline that circulates in blood, or I should say most phosphatidylcholine, or phospholipids, are synthesized de novo. And this was what made create the initial idea that perhaps dietary choline that's present in feed ingredients would be degraded by the gastrointestinal microbes into this oxidized trimethylamine product. Therefore, it would limit the supply of choline to synthesize phospholipids. And eventually, this was confirmed by Dr. Rich Erdman here at the University of Maryland in the United States, that showed that dietary choline or choline salts such as choline chloride is extensively degraded by the rumen microbes, and less than 10% of the dietary choline actually reaches the small intestine in dairy cows. And then Dr. Erdman went on to do experiments either feeding or infusing in the abomasum choline, and he showed that responses were obtained only when he infused choline salts into the abomasum, reinforcing this concept that choline is extensively degraded by the rumen microbes and therefore requires protection against microbial metabolism. So upon the development of rumen-protected choline products, multiple universities in the United States, in Italy, in the Netherlands, and other places showed that, in fact, supplementing choline in the form of rumen-protected choline benefit transition dairy cows, as it does for growing ruminants as well as other species. And I will spend a little bit of time showing what those benefits are in this presentation. So choline is a required nutrient, but obviously phospholipids are essential in the diet of ruminants, or they are essential for cell survival. But choline also play important roles in the structural integrity of cells because it's part of phospholipids. It plays important roles in neural tissue. So choline, when it's acetylated, produces acetylcholine, which is a neurotransmitter. Choline also plays an important role controlling gene transcription because it can be oxidized to betaine, and betaine is a methyl donor that can transfer methyl groups to resynthesize methionine, and that can be converted into S-adenosyl methionine, which is the universal methyl donor. And methylation or transmethylation reactions can control how genes are expressed in cells, and this can have an impact on protein synthesis. But also, probably choline is better known for its role as a component of phospholipids, which is critical for the absorption and transport of lipids in aqueous solutions, such as the synthesis of lipoprotein for absorption of fatty acids in the gastrointestinal tract by the role of chylomicrons, the transport of fatty acids as lipoproteins that can be delivered to the mammary gland, in the case of ruminants, primarily through LDL cholesterol, HDL cholesterol, and also the role that choline plays in export of triacylglycerols that have been resynthesized in the hepatic tissue by the role of very low density lipoprotein. So if you look at literature, if you think of a human being such as Dr. Clay Zimmerman here, in the human literature, depending upon whether you're a woman, if you're pregnant or not, or if you're a man, and if you're an adult man, the recommended daily allowance for choline are anywhere between 400 to 550 milligrams per day, so approximately half a gram. If you look at the poultry literature, laying hens, based on their dry matter intake, they require approximately the same amount because there is a large amount of choline or phospholipids that get transferred to the egg. A young chick requires less, about 100 milligrams per day. A lactating sow, depending upon her dry matter intake, she requires 4 to 6 grams of choline in the diet. Obviously, for all of these species, it's a lot easier to supplement choline because we can provide as choline chloride or another salt because we don't have this extensive pre-duodenal degradation of choline. Therefore, the ability to supply choline for absorption and transport in the small intestine is much more effective. But the issue becomes when we go to ruminant animals, lactating dairy cows, for instance. So now we have the impediment of the rumen, and I think this is probably the major reason why we haven't established recommended amounts of choline in the diet of adult ruminants, because there is a challenge here. First, we have to protect choline in some sort of matrix, most commonly in a lipid matrix, to prevent degradation by rumen microbes. And then we have to have some prediction of how much of that choline becomes available to be absorbed in the small intestine. So those limitations probably precluded the establishment of recommended amounts to be fed for cattle. And perhaps another reason is that we don't have strong experiments, or experiments, I should say, showing those responses in terms of lactation performance. I'll show you extensive literature that we do have, and I think we know the benefits that we get from supplementing rumen-protected choline, but we don't have what is the optimum amount to be fed at this point. And that probably has precluded people from saying we need to feed so many grams per cow per day. But I hope to make the case here today that we should feed to every cow during the transition period. So here is the classic pathway for choline metabolism in tissues when it is absorbed from the gastrointestinal tract. So choline, when escapes microbial degradation and it's absorbed, it can be acetylate in neurons, whether it's in the central nervous system or in the gastrointestinal tract or in other tissues, to synthesize this neurotransmitter acetylcholine, which is important to maintain nerve transmission or transmission of a nerve impulse in the neuromuscular junction. Choline can also be reversibly oxidized to betaine, and betaine is a methyl donor. Okay, so betaine can donate methyl groups to homocysteine to regenerate methionine through the action of this enzyme and methionine synthase. And there is an interplay here between choline and B vitamins such as folic acid. Methionine now can be converted into S-adenosyl methionine, and this can recirculate components of methionine such that you can regenerate the amino acid. Okay. However, if choline is absorbed, it can be phosphorylated into phosphocholine through the Kennedy pathway. And this pathway is present in almost every cell that's being studied. And this is part of the synthesis of long-chain fatty acids, the Kennedy pathway, or triglycerides, I should say. Then phosphocholine can combine with CDP, with CTP first, cytidine 3-phosphate, and eventually can become cytidine diphosphate choline, and eventually it can become phosphatidylcholine, which is the main phospholipid in ruminants. But it can also be synthesized from phosphatidyletanolamine through transmethylation reactions by methyl groups that are donated by S-adenosyl methionine. So CDP etanolamine can receive 3 methyl groups from S-adenosyl methionine, and some of those methyl groups can be originated from betaine, which is the product that was originated from oxidation of choline. So you can see that we can have de novo synthesis of phosphatidylcholine from dietary choline through phosphorylation, or we can have de novo synthesis of phosphatidylcholine that's specific to the liver tissue through transmethylation reactions of phosphatidylethaноlamine. And there are multiple enzymes and compounds that play a role in this process. But choline plays a central role in both pathways for synthesis of phosphatidylcholine. Now, if we go back to our transition dairy cow, so as parturition approaches, we know that particularly in the last week of gestation, the combined effects of the exponential growth of the calf with the increased uptake of nutrients associated with the synthesis of colostrum by the mammary gland increases the nutrient needs of the cow. But unfortunately, this cow has inherent reduction in dry matter intake. Most cows will decrease dry matter intake as they get close to calving. So the association between increased nutrient needs, reduced dry matter intake, and the signals to initiate lactation induce a marked increase in lipolytic signals. And this induces increased flux of fatty acids in the circulation. So lipolysis is upregulated, and that is an adaptive mechanism to initiate lactation. It's perfectly fine to occur. So those fatty acids can be taken up by tissue, such as the muscle tissue, can be taken up by the mammary gland, okay, to synthesize milk fat once lactation initiates. But it can also be taken up through the arterial circulation by the hepatocytes. And the liver will use those fatty acids to synthesize energy because now hepatic metabolism is upregulated because the cow has to increase synthesis of nutrients to supply the mammary gland. The issue becomes when the influx of fatty acids is in excess of the energy needs by the hepatocyte. Then the hepatocyte uses other routes to dispose of that excess of carbon, one of which is ketogenesis, which is basically an adaptive process during periods of inadequate nutrient intake. And these ketones now can be exported to the general circulation. They are water soluble. So the main one that we typically think of is beta hydroxybutyrate. And this can be used as a substrate for de novo synthesis of fatty acids. However, when the influx of fatty acids is excessive, or for some reason the liver is unable to process them properly, they accumulate and they will basically be esterified within hepatocytes and they will be deposited as lipid droplets. And these lipid droplets are quite dynamic. They are not just storage of triglycerides. They actually secrete molecules. And animals will develop what we call fatty liver or hepatic liptosis. Part of the reason why ruminants are sensitive to hepatic liptosis is because, as opposed to monogastrics that have the great ability to export these triglycerides as lipoprotein rich triglycerides such as VLDL, ruminants evolve without having to have a hepatic tissue that is lipogenic. Therefore, it never really had to secrete large quantities of VLDL. So the ability to export triglycerides from the liver in ruminants is inherently less than what's observed by hepatocytes in monogastrics, meaning that it just takes time. It takes longer in ruminants to dispose of the excess of fatty acids that have been accumulated as triglycerides in the liver. And this accumulation can damage the cell, can induce pro-inflammatory response, and can induce the production of acute phase response. Obviously, this diverts nutrients from productive synthesis, productive purposes, because now we're building an inflammatory response that not only can damage the tissue and affect tissue function, and people have shown decreased ureagenesis, decreased gluconeogenesis when there's excess of triglycerides in the liver, but also now uses nutrients to synthesize anti-inflammatory molecules, which will divert nutrients from milk synthesis. Now, VLDL that is exported from the liver can bring nutrients to the systemic circulation, and some of that can be delivered to the mammary gland. Okay, but we know that during early lactation, when cows are more susceptible to fatty liver, at this point, the supply of nutrients is limited, one of which is methionine, a essential and required amino acid, as you probably just heard from Dr. Mike Van Amber. Now, because the supply of methionine relative to the needs is limiting, and if we go back to that pathway of methionine being a methyl donor or being a precursor for S-adenosylmethionine, which is a methyl donor, we can expect that the supply of methyl groups for synthesis of phosphatidylcholine through the PEMT pathway would probably be limited during this stage of lactation, because now the cow is using this amino acid to synthesize protein in colostrum and milk. So at this stage, very likely cows become more dependent on the phosphorylation of choline, so long choline is available to synthesize phosphatidylcholine. And when animals have inability to process triglycerides, eventually they can develop fatty liver, like in this extreme case of a cow that had excessive hepatic lipidosis. So how common is this problem? It's actually quite common. Hepatic lipidosis affects almost half of the cows that we can observe in the literature. So if you look at moderate degree of hepatic lipidosis, at least 5% triacylglycerol, it affects almost 40% of the cows. If we combine moderate with severe, you almost have 50% of the cows with hepatic lipidosis between the first and second week of lactation. And it takes several weeks for this process, or for hepatic triacylglycerol to decrease to the typical values, which are 1 to 2% on a wet basis of the hepatic tissue. Now, what are the consequences of hepatic lipidosis? So we recently addressed this by looking at data from a little over 330 cows, in which we had detailed information, and we collect liver tissue from those animals at approximately 8 days postpartum. And we just asked the question, is there an association between hepatic triacylglycerol with intake, yields of milk, yields of energy-correct milk, and health of cows? So let me show you some of those responses. So in our statistical model, we use triacylglycerol as a continuous covariate in the model. Okay? As opposed to classifying an animal as having or not having hepatic lipidosis, we ask the question, at what point of hepatic triacylglycerol we affect lactation performance. And what we found was this quadratic association between them. So when concentrations of hepatic triacylglycerol increase up to about 4 to 6%, production increase. And after that point, production decrease. Whether we looked at milk yield or energy-corrected milk yield. Each triangle here represents one or more cows, depending upon if they had exactly the same triacylglycerol or not, and exactly the same response, milk or energy-corrected milk. So we can see that there is this quadratic relationship that up to a point, there is no negative association, but after 4 to 7% triacylglycerol, then performance begins to decline. Then when we looked at incidence of disease and survival of cows, the response was slightly different. Now what we found were linear associations, no longer quadratic associations, meaning that as the concentration of hepatic triacylglycerol increase, in the case of metritis, the risk of metritis also increased. And you can see here, a cow with 2.5% hepatic triacylglycerol had 12% incidence of metritis, and this incidence doubled when hepatic triacylglycerol was 7.5%. If we just look at morbidity, we can see that there was a 10 percentage point increase in the diagnosis of disease in the first 90 days of lactation. And there was a tendency to increase culling and death. So animals that left the herd by 300 days in milk, we had 5 percentage points more when they had excess of triacylglycerol. In excessive concentration of triacylglycerol in the liver in the first week that was associated with increased hyperketonemia and also increased hypocalcemia. So we can see this typical associations between disease in cattle that result in increased losses, whether they are production or they are related to health of cows. Now, one of the important concepts is that as cows approach calving and initiate lactation, the concentration of phospholipids in plasma is the smallest observed during the transition period. And we think that this very low concentration of phospholipids in plasma is related to the supply of building blocks for phospholipid synthesis, concurrent with the uptake of phospholipids by the mammary gland and the uptake of, for example, methionine that could be used to donate methyl groups for synthesis of phosphatidylcholine. And this makes the case that probably this is the target time to supply choline in a protected form to lactating dairy cows. Now, choline plays an important role in phospholipid synthesis. So these are data from cows that had a low concentration of hepatic triacylglycerol, or cows that were classified as having fatty liver. And these are proteins that are part or components of that lipoprotein called very low density lipoprotein that's required for export of triacylglycerol. This is a protein that carries triglycerides to be packaged into the organelles of the hepatocyte. And these two proteins here are components of VLDL. And we can clearly see that this microsomal transfer triglyceride protein is reduced in abundance in cows that develop fatty liver. Apolipoprotein B100, which is the main apolipoprotein of VLDL, is also reduced in cows that were diagnosed with fatty liver, and the same is true for apolipoprotein E. So cows that have fatty liver, they are associated with a reduction in this machinery to synthesize and package VLDL to export triglycerides. And in the monogastric literature, when choline is supplemented, which is in this case here compared to choline deficient diets, we can see that there is an increase in chylomicron assembly based on an increased concentration of apolipoprotein B48. This is the main apoprotein present in chylomicrons during the process of absorption of fatty acids and export of triglycerides into the lymphatic system. So we can see that absorption of fat can be limited when animals are deficient in choline, at least based on the monogastric literature. And supplementing choline enhances that. Now, choline has lipotropic effects. In other words, has this ability to facilitate transport of lipids from one tissue to another. So these are the initial indications that choline reduces the risk of hepatic lipidosis in cattle. This is work by Dr. Rick Remmer when he was at the University of Wisconsin, in which he showed that after induction of fatty liver in cows, animals that were supplemented with choline, they had reduced concentrations of triacylglycerol in the liver compared to untreated controls. So these cows were induced to develop fatty liver very successfully, and you can see that the increase in hepatic triacylglycerol was much greater in the control than in the cows that were fed rumen-protected choline. We replicated this experiment titrating the dose of choline. In this case, I'm showing here is the amount of choline ion as choline chloride that's been encapsulated in a rumen-protected form. Okay, and you can see that as we increase the intake of choline ion by these cows that were induced to develop fatty liver, the concentration of hepatic triacylglycerol linearly decreased. This is a decrease of approximately 40% in hepatic TAG. So the cow responds exactly like monogastrics, supplying choline in periods in which they are susceptible to fatty liver. Choline attenuates the degree of hepatic lipidosis. Okay, in the data from Marcus Zenobi, we recently looked at what genes are altered in the hepatic tissue that perhaps explain some of the differences in hepatic triglyceride. And interestingly, we found, so let me just explain this here. This is the dose of choline ion on the bottom of this heat map. So think of this as the x-axis of a graph, okay? And then every row here represents a gene, okay, that's expressed in the hepatic tissue. And as I go from red to green, the abundance of that gene or the abundance of message for this particular protein increases. If I go from green to red, it decreases. And interestingly, what we found was that genes involved in antioxidant defense, synthesis of VLDL, transfer of triglycerides, or activation of choline, they were all upregulated as we increase the amount of choline fed to cows. On the other hand, genes involved in synthesis of triglycerides, production of pro-inflammatory molecules, they were downregulated in animals as we increase the supply of choline ion in the form of rumen-protected choline. So this is suggesting to me that by supplying choline, the hepatocyte changes how it works, and that affects either because of choline itself, or in this case, perhaps because of the reduced triacylglycerol concentration. It alters how it expresses genes that can affect the production of proteins that are important for export of triacylglycerols, that is important for antioxidant defense, and minimizes reesterification and synthesis of triacylglycerol, and minimizes the pro-inflammatory response in the hepatocyte. Okay, so we decided to go back and review those data and replicate that experiment with a slightly different approach. And I'll show you a series of experiments now that we just completed, and they're in process of submission for publication. So in this first one here, we subject 110 cows to a fatty liver induction protocol, in which in the first few days, we basically replicate what Dr. Zenobi and what Rick Grammer did in their experiments. So we had cows that were fed at Lipton for 5 days, and then we had a period of negative nutrient balance because we restrict the amount of feed offer and net energy for lactation offer to those cows. So basically what we did is we calculate the net energy needs for maintenance and pregnancy, and during feed restriction, we offer those cows only 50% of that. We use, at the time, we used the previous NRC values for maintenance, and we know that the new nascent dairy calculates a larger value for maintenance. So now it's 100 kilocalories of net energy per kilogram of metabolic weight. So based on the new NRC, we induce even a more severe negative nutrient balance during this period. And then we apply treatments here, and you will see what the treatments were. And we measure things throughout this experiment in those cows. These were cows that were 230 days of gestation, so they were dry cows. And we collect liver tissue here, right at the end of the ad libitum feeding period, and then at the end of the feed restriction to measure triacylglycerol, glycogen, and other things. And we collect blood at several time points. So the treatments were a negative control, no supplemental choline, then 12.9 grams of choline ion. This is the standard dose that most people would feed in transition cow diets, of a product that has a low concentration of choline chloride. Then we increased the dose using the same product. And then we fed the same doses, but in a product that has a greater concentration of choline chloride. So what I want to do here is show you, because of sake of time, the effect of choline, which is the contrast. We applied what's called orthogonal contrast to compare this. which is the effect of choline against control. And then I want to focus on the dose of choline, which is at 12.9 versus 25.8, the main effect of dose of choline. And I'm going to neglect the other response just because we don't have time to go through every piece of data, but I'll be glad to cover this if we have questions. So here's just to show that obviously we underfed cows. So in a period of 9 days, these cows were losing 2 kilograms per day. And with that, they lost body condition. So we induced extensive lipomobilization in those cows. You can see here is the blood NIFA of those cows, 0.28 millimolar when they were fed at Lipton. And during feed restriction, we greatly increased to 1.1 millimolar, typical of a cow that's losing a lot of weight, which is illustrated here. That would be compatible to a transition cow. And with that, ketogenesis was increased. So we increased concentrations of beta-hydroxybutyrate during feed restriction. These are responses that we were expecting. And as we can see here, there are no effects of treatment. So whatever I'm going to show you from this point on is unrelated to the level to the degree of lipomobilization because they did not differ. So here's the hepatic composition. First, how much did cows eat during the feed restriction? They were eating only 5 kilograms of dry matter, so approximately 11 pounds. It's no surprise that they were in extensive negative energy balance, 10 megacalories of net energy for lactation, negative. So this should induce hepatic lipidosis, and in fact it did. On day 13, this is the concentration of hepatic triacylglycerol in the control cows, 9%. On a dry matter basis, 26%. And we can clearly see that as we feed choline, it greatly reduces hepatic tag. That's the first point. And as we fed a larger dose, it further reduced hepatic triacylglycerol. As you can see, going from 12.9 to 25.8, further reduced, exactly like Dr. Zenobi showed. Also, we can see that concentration of glycogen in the hepatic tissue increased with feeding choline. And again, the increment was greater when we increased the dose of rumen-protected choline to those cows, such that if we look at the ratio of triacylglycerol to glycogen, it was a lot more favorable in cows that were supplemented with choline compared to control cows. And it was even more favorable when we increased the dose. So basically what I'm showing you here is one more time, choline has this lipotropic effect. It reduces the risk of hepatic lipidosis, and as we increase the dose, the benefits increase in this cow that is induced to have hepatic steatosis. We were quite excited about this data because when we analyzed aptoglobin, we saw that cows that received choline, despite of the fact that they had a small increment in hepatic triacylglycerol, we did not see any increment in concentrations of this acute phase protein. Whereas in cows that were in the control treatment, there was a 50% increase or 60% increase in the concentration of aptoglobin. So this tells us that probably that large influx of triacylglycerol is inducing a pro-inflammatory response, and the liver is responding by upregulating aptoglobin synthesis. Or perhaps the supply of choline that's being fed to cows is increasing the amount of phosphatidylcholine, and phosphatidylcholine is known to have anti-inflammatory properties. So one of those things are happening here, or is happening here. Now, we went on to complete another experiment, and this is probably the key response that we want to address, is can we alter secretion of triacylglycerol-rich lipoprotein? In other words, VLDL secretion by the hepatic tissue by supplementing choline to cows that are induced to develop fatty liver, the same way that choline has been shown to do this in other species. Okay, so what we did here is we enrolled 33 cows, and they were assigned to one of three treatments: a control or 25.8 grams of choline ion in one of two sources. But again, just for the sake of time, I want to compare control against 25.8 grams of choline. And what we did after inducing fatty liver in cows and feeding this for 13 days, we gave a dose of this non-ionic detergent called thiloxapol IV, and this coats the lipoprotein particles. It coats these lipoprotein particles and prevents lipoprotein lipase from hydrolyzing the triacylglycerols that are contained within these lipoprotein particles. So by doing this, we induce hyperlipidemia. So the concentrations of lipids in blood go up. And one of the lipids that increase in concentration is triacylglycerol. And most triacylglycerol in blood of cows originates from VLDL secretion from the liver. So we basically collect sequential blood samples, and then we collect also lymph to characterize the metabolome of lymph. So I'm going to compare control against 25.8 grams of choline ion. Okay, so these are the concentrations of triacylglycerol in serum of cows in the control treatment or cows receiving rumen-protected choline. And we can clearly see the concentration of triacylglycerol increase over time, but the increment is greater in animals that receive rumen-protected choline. And you can see here the interaction of treatment by time. So when we measure the area under those curves, this is what we found, that cows that received rumen-protected choline had greater accumulation of hepatic triacylglycerol, and this accumulation is because of nascent hepatic triacylglycerol that's being secreted by the liver, and this strongly suggests that hepatic secretion of triacylglycerol, in other words, the export of VLDL, is enhanced in cows that are supplemented with rumen-protected choline compared to controls, exactly as it's been shown in rats, guinea pigs, mice, and dogs in the literature. So one more time, the mechanisms that are observed with supplemental choline in the diet of monogastrics can be replicated in diets of lactating dairy cows, so long we supply choline for absorption post-rumen. Then within this series of experiments, we fed cows diets to induce fatty liver, and then we supplement them with the same treatments, control or a high dose of choline ion as rumen-protected choline. And then we, in the last day of the experiment, on day 14, we made those cows be off feed, and we dosed them with 450 grams of long-chain fatty acids, primarily palmitate was fed to those cows. So we made sure that they ate everything, and then we collect serial blood samples, but then we collect lymph from those cows, and that's what I'm going to show to you. So this is the composition of lymph. We assayed over 500 metabolites in lymph. Of those, 400 and some we had reliable data. This lymph was collected from the superficial udder lymph vessel that's in the posterior portion of the udder. And as we go from blue to brownish reddish, we see an increase in concentration. And you can see the clustering of animals. There are 30 cows here. There are 10, if I recall, 1, 2, maybe fewer than 10, just because we didn't get lymph from 3 animals in this experiment. But you can see the reds represent the control cows. The greens represent each of the cows that receive rumen-protected choline. As we go from blue to the dark color, the concentration of the analyte measured here increased. Okay, so we can see that as I go from the right to the left, I go from cows that receive control to cows that receive rumen-protected choline. And we can clearly see an increase in concentration of the analyte. So what were the metabolites that changed? There were over 20-some analytes that changed in the lymph. Here are some of them, the ones that change in the greatest amount. And you can see that they were diglycerides, they were triacylglycerol, they were choline and amino acids. So you can see that by supplying rumen-protected choline in the diet of those cows, we increase the concentration of choline in the lymph tissue. Perhaps because we increase absorption of choline, or we increase synthesis of phosphatidylcholine in chylomicrons, and then some choline is transported together with those chylomicrons in the lymphatic fluid. Well, I hope I made the case that choline is lipotropic, and the mechanism is very similar to that observed in monogastric species. Now I want to spend a little bit of time showing what are the benefits to lactation when cows are supplemented with choline. So a few years ago, we put together all the literature we could find on randomized experiments in which cows were fed a controlled diet or a diet supplement with some amount of rumen-protected choline, starting prepartum and continuing postpartum, so during the transition period. So what we found were 22 experiments in 21 published manuscripts that compile 30 treatment means in which the amount of choline was 0 grams per day, and 36 treatment means in which the amount of choline fed was anywhere between 5 to 25 grams of choline ion in a rumen-protected form. So here you can see the histogram of the amount of choline ion that was supplemented to cows in the transition period in the treated groups. And you can see that the vast majority of the treatments had this intermediate concentration or amount of choline ion. Most experiments, no surprise, had 12.8, 12.9 grams of choline ion as rumen-protected choline supplemented to cows. But there were some that fed as much as 25 grams, as you can see here. So what we did is we asked the question, what is the effect, in this case, because in every experiment we had a negative control, what is the effect of manipulating the amount of rumen-protected choline on postpartum performance and health of cows? So for that, we collected diets that were fed to those cows. And one of the questions we want to address is there a relationship between the supply of choline and supply of methionine? So we enter every single diet, adjusted for the observed intake, and we calculate the supply of metabolizable methionine prepartum of those cows, either as grams per day or as percent of the metabolizable protein supply in the diet. And we did the same postpartum. So if we look at prepartum, most diets were between 1.9 and 2.2. And this was calculated using the 2001 NRC, which probably results in a little less metabolized methionine values than if you were to use the CNCPS or the AMTS model and the AMTS software, the NDS software that use the CNCPS biology program. And here's the postpartum. You can see that most diets were between 1.7 and 2.4, and there were a few diets that had a lot of rumen-protected methionine supplements, so they had values that were in the 2.7, 2.8% of the MP as metabolizable methionine. Okay, so let me quickly go through this. This is energy-corrected milk response. Every row represents a comparison against the negative control. The names here indicate the reference of the paper that originated that. So you can see that in Hartwell, in Indiana, they had several comparisons against the control because they had different combinations of treatment. Experiment in Cornell by Tom Overton, they had increased doses of choline ion fed to cows. So we compared everything against the control. Okay? So here's the central line that would indicate exactly the same response, okay, between treated and control. Every time they estimate the dark dot falls to the left, it would indicate that the response, in this case, energy correct milk, decrease in the treated compared to the control. Every time the point estimate falls to the right of the central line, would indicate that the point estimate is a positive value, therefore the response increase. In other words, energy correct milk would increase. The size of the gray square represents the weight of that comparison in the meta-analysis. Okay, so this is a weighted value. And the horizontal line represents the 95% confidence interval. Okay, so in this forest plot, we can see how consistent the response of rumen-protected choline is across all these experiments that have multiple diets, multiple types of cows, multiple environments. And we can clearly see that there's a lot of external validity. In other words, supplementing choline to cows during the transition period as rumen-protected choline increases energy-corrected milk. What is the increase that we observed? It was 0.38 standard deviations, which is approximately 2.2 kilograms per day. Okay, so 4.4 pounds per day increase in production that was consistent. Most cases it increased. However, the response was heterogeneous. So there are things that influence the level of response of those cows. So we decided to investigate that. And one of the things that we investigate was dose of choline. And you can see here that as cows consume more choline, this line here, the central line, is the overall response from the statistical model, the mixed model that we use for this meta-analysis. And you can see that there was an increase with dose of choline, and this increase was linear. Okay, so when cows received 12.9 grams of choline ion, the increment in milk yield was of 1.6 compared to the non-supplement cows. If we look at energy-corrected milk, the increase was 1.7. But you can see that in both cases, we had an interaction with metabolized methionine postpartum, meaning that the response to choline was dependent on the level of metabolized methionine in the diet postpartum. So we decided to explore that. And this is what we found, and I hope I can make this figure clear. So these are cows, this is the prediction of milk yield of cows that received 0 grams of choline. Okay, so let's just compare the 0 against the 12 grams of choline ion, just for easy of displaying these results. When metabolized methionine postpartum was low, 1.7, we can see that these cows respond with increased milk yield, and this response was quite substantial. When the supply of metabolized methionine diet postpartum was quite high, 2.3 to 2.4, they also respond in increased milk yield with supplementing 12 grams of choline ion, but that response became smaller, but still positive. Okay, so at 2.3, the response was 1 kilogram of milk in the postpartum period. Now, what about energy-correct milk? We see exactly the same pattern, okay, this interaction. At very low metabolized methionine, we have a superb response, several kilograms of milk. At very high metabolized methionine, we still see a response. It becomes smaller, but still positive. So this is telling me that although there are relationships between choline and methionine, there are additive effects of supplying both in the diet of transition cows. So cows benefit from both, because we know that formulating for metabolized methionine or formulating for amino acids is important, particularly in the postpartum period. And then we ask the question, what is the impact of supplementing rumen-protected choline on feed efficiency? So this is the same type of graph, the forest plot, and the overall response was 60 grams more energy-correct milk for every kilogram of dry matter intake these cows consumed across all of these experiments that had both energy-correct milk measure and dry matter intake measure at the individual cow. So finally, we looked at health of cows. I want to point out that not every experiment displayed the effects of choline on health. Many people don't report health effects just because they have a small experiment. And with that, we are unable to compile those data. So out of the 21 experiments, you can see that only 10 to 12 experiments report health incidents. But we can also see here that there were indications that supplementing choline reduced the risk of some disease. One was retained placenta, the other one was mastitis in this particular case here. So cows that were fed choline as rumen-protected choline had smaller incidence of those two diseases. I want to touch on two other important points. In two experiments that we completed here at the University of Florida, we saw this interesting phenomenon. Okay, we supplemented cows during the transition period without or with rumen-protected choline. So from 3 weeks before calving, starting at approximately 255 days of gestation, up to 21 days postpartum. In these two experiments, cows either received choline or they did not receive supplemental choline. Okay, but we measure production for either during a period of 40 weeks or 25 weeks. And despite of the fact that we stopped supplementation here at 21 days postpartum, cows continue to produce more milk when they were supplemented with rumen-protected choline. So in this first experiment here by Marcus Zanobi, an increment of 2.1 kilograms. In the second experiment here by Juan Bolatti, an increment of 2 kilograms up to 25 weeks. And I think this is a common finding that when we do good things to cows during the transition period, they respond with additional benefits above and beyond the period of intervention. And that's exactly what we found here. And I think this goes back to the fact that cows during the transition period have limited concentrations of phospholipids. And perhaps supplying rumen-protected choline provides substrate for more phospholipids. And we know that at this time, we have remodeling of the mammary gland. We have replication of mammary cells, active mitotic activity in the mammary gland. And obviously, cells require phospholipids. And there's this interesting concept in the human literature that this enzyme here, choline kinase activity, is greatly increased at the onset of lactation and also greatly increased during periods of cancer, mammary cancer. It's actually used as a marker for cancer in humans. And this is the enzyme that activates choline to phosphocholine. So perhaps by supplying choline at this stage during the transition period, we upregulate the activity of choline kinase to produce more phosphocholine, which is incorporated into the mammary tissue for synthesis of new cells that will be secretory cells during the remainder of the lactation. And this perhaps explains part of this increased response during lactation because of this mitotic activity that takes place during transition and the need for phospholipids to be incorporated into that mammary tissue. Lastly, I want to touch on this concept that every cow benefits from choline, and this dogma that we should feed choline only to cows that are high risk of fatty liver. So the way we address this is we ask the question, does the body condition score when cows enter the prepartum group influence the response to choline when they are supplemented during transition? So we pulled data from 233 cows from two experiments, and we basically asked, does body condition score during the transition period affect response to choline during the subsequent lactation? So here's the distribution of body condition score in cows that were not supplemented with choline or supplemented with choline in Zanobi's experiment, and the same for Bolatti's experiment. You can see that we have a range from low body score to excessive amounts of body condition when cows entered the prepartum period. So here's the distribution of body condition score. We have cows that have low, moderate body condition. We have cows that are over-conditioned, whether they receive control or they receive choline. Okay, so this is, I'm going to show you only two responses, milk yield and energy-corrected milk yield. Each dot represents a cow here. And here are the results of the statistical model. And you can clearly see that there is an effect of rumen-protected choline, which is the difference between the blue and the orange line. Okay, those are the estimates from the choline. In other words, this is your least square means that you see typically in publications or in a table. But you also see no interaction between rumen-protected choline and body condition score when cows enter the transition group, meaning that regardless of a cow that is 2.75 or 4.0, supplementing rumen-protected choline increase milk and increase energy-corrected milk. I'm going to remove the dots so you can see the lines, and you can clearly see that there is an increment independent on body condition score. So what I mean by that is a skinny cow or a fat cow, they both respond with increased milk and energy-correct milk. And I think there are many reasons for that. Okay. So, and that's where I want to finish up with this potential mechanism. There are things above and beyond this role of choline on fatty liver that might explain differences in production. So obviously the first one is that cows are no different than other species. Supplementing rumen protect choline enhances triacylglycerol export from the liver, and that's what I showed you from the work of Usman Arshad that will be soon submitted for publication. I also showed you that supplementing choline to choline deficient diet enhances apolipoprotein B48, which is a marker for chylomicron. It's a component of chylomicron, and chylomicrons are responsible for transporting lipids that are absorbed in the gastrointestinal tract into the lymphatic system. Cows seem to be no different. When Marcus and those cows with fat, those that had received choline during the experiment, they had increased plasma concentrations of triacylglycerol compared to control, suggesting that probably absorption of those lipids is enhanced. Now, choline is needed for synthesis of phosphatidylcholine, and we have heard a lot of data recently, particularly from Lance Baumgart's group, that when cows undergo transition, they are subject to or susceptible to leaky gut. And deficiency of choline in monogastric species induces leak gut. So it is possible that by supplying choline, we provide more phosphatidylcholine that's important for integrity of the intestinal lining. That's one aspect. The second one is that phosphatidylcholine has marked anti-inflammatory response. It dampens the inflammatory response, which can be beneficial for the liver, but also can be beneficial for the gastrointestinal tract. Choline, as you remember, is part of this one carbon metabolism. And there's some very cool data showing that in mice, unfortunately not in cattle, that when they breed mice to have microbes in the gut that are unable to degrade choline into these 3-methylamine oxides, the offspring of those animals, so you preserve choline by doing this, okay? So you breed mice to have a microflora in the gut that is unable to degrade choline in these 3-methylamine oxides. Therefore, you have more choline that provides a substrate for betaine production, and betaine can affect transmethylation reactions. When they evaluate the pups of these animals, they show that there is increased DNA methylation and decreased lots of issues in this new generation of animals, such that animals that were provided with more choline during gestation have reduced adiposity, less metabolic disease, and less and reduced changes in behavior such as anxiety. Lastly, I just want to finish with this idea here that this enzyme, choline kinase, that phosphorylates choline into phosphocholine, is upregulated during the onset of lactation. And perhaps by providing more substrate, we either affect the enzyme expression or the enzyme activity, and now we have more phosphatidylcholine that can stimulate mitotic activity in the mammary gland, that can have this effect on proliferation of mammary cells, and eventually this might explain this extended benefit in terms of lactation performance when we supplement rumen-protected choline to cows during the transition, but that benefit extends above and beyond the period of supplementation. So I hope that during my presentation I made the case that it's unquestionable that choline fits the criteria of a required nutrient for dairy cattle. Okay? When we supplement choline as rumen-protected choline, we reduce triacylglycerol, exactly like people have shown for the last 100 years or 90 years in the monogastric literature. But choline also has impacts on intake postpartum. I didn't show you this data, but in the meta-analysis we showed some increment in intake. But I clearly showed to you that there is a consistent increase in milk, energy-correct milk, and milk components. And also it tends to reduce the incidence of some disease. So I think this would fit the criteria of a required nutrient. It improves animal health, stimulates lactation, and when deficient, it can increase the risk of a disease such as hepatic lipdosis. Okay? The response to rumen protect choline are influenced by the supply of metabolizable methionine. However, they seem to be additive. Both of them would stimulate lactation performance. The response or the benefits to rumen protect choline, as opposed to what many believe, is observed or they are observed regardless of degree of fatness of cows. So whether a cow is skinny or whether a cow is fat, they both respond with improved lactation performance in the postpartum period. And the benefits go above and beyond the period of supplementation. So lastly, what are the mechanisms? Obviously, I showed you the impact on triacylglycerol, but perhaps choline, by providing substrate for phosphatidylcholine synthesis, may increase nutrient intake in these very early stages of lactation, may improve gastrointestinal integrity. It improves health, reduces hepatic lipidosis, and has indication of reduced risk of clinical disease, but it may also have other non-classical mechanisms such as reduced inflammatory response, and there's good data to support this, and perhaps it also stimulates memory cell proliferation. So I really thank you for your attention. I wish I were there, present at the Cornell Nutrition Conference. Unfortunately, I wasn't able to, and I hope that there will be some discussion either now or at the end of this pre-conference. Thank you very much.